So now all these things I told. So these are all the things stepwise we can approach the repeat bleeding. But if it is not conceiving, then what it can be? So there comes this one, because 29% of the animals are infected with subclinical, and even 30 to 40% of the repeat bleeding, they do have subclinical uterine infection. So in that case, the importance of detecting subclinical uterine infection in repeat bleeder assumes a larger role. Why? Because of this. Again, like mastitis, what we thought over there, it was disease of udder. It is not so. So similarly here, uterine infection, what we thought is it is restricted to the uterus. It is not so. If uterus is affected, uterine infection is there, be it a subclinical or clinical or beyond. So in that case, all these activity, including the LH secretion, including the estrogen production, and then the cyclicity, everything is going to be altered. And in that case, even if the animal is subclinical uterine infection, which is not clearly visible from outside, all these changes are already taking place in its body. So that's how it culminates into delayed ovulation or aging of gametes or embryonic mortality. So ultimately, it has to be contained. So when we talk about uterine infection, these are all the commonly asked, very commonly asked question. First and foremost thing is, tell that subclinical infection is causing a devastating effect on the reproduction. But how to diagnose? There is no tool at field condition to diagnose. Yes, there are no kits, but there are some methods that will be available that I will be explaining. What is the best treatment protocol? You and me cannot decide, but there are some set of protocol that is suggestive of designing a treatment protocol. It is not a thumb rule. No antibiotic can be flat outly recommended for all the animals across the country. Should intrauterine treatment be given or not? It's also one question. And why some animals do not respond to antibiotic treatment? That is also $1 million question. And how to prevent bovines from that getting uterine infection. Understand that how the uterine infection develops. 100% of the cows or buffaloes after calving, they harbor microorganisms. That is a universal truth. Not a single animal is free from infection or free from microbial contamination at the time of calving. Yes. So what happens then? Almost 90% in a well-managed head and 60% in the average managed head, they themselves clear the microbial load or excrete all the microbial organisms. The remaining persons, they carry and they develop into different forms of uterine infection. Why this happens? It is because of this. Because it is not only the host. It is an interaction between the host, environment, and the agent. How virulent the agent is, how much pathogenic it is, and then what is the number of pathogens that is invading the uterus. That decides the development of the uterine infection. And the host, as I told earlier, unless and otherwise it is fed properly. The nutrition is the most important. If you want to get a good reproduction, without nutrition, it is not possible. So first we have to take care of the nutrition, immunity, and less stress at that time. Then the immunity, then the animal will be able to clear herself all this microorganism contamination. And then the environment, it varies with the management system and the farming conditions. If it is more number of animals, it is totally different. But however, it should be housing should be hygienic. If it is not, all these three gets into development of the uterine infection in the cows. It is just a balance between the immunity, bacterial pathogenicity, and the environment. So there are some risk factors which we can identify and which we can reduce. These are the things that we conducted a study in NDRI. When I was there in NDRI Karnal, used thousands of the animals. So if you see, I want to highlight only one point. There are many factors that contribute to development of uterine infection in the latter part. But one thing is very important is retention of fetal membrane. Once the retention of fetal membrane, the number which I have given here is odds ratio. For example, if a crossbred animal, which is normal cow and which had RFM, the animal which had retention of fetal membrane 100 times more at risk of developing uterine infection. That was, it says there is a risk. So similarly in Jebu, similarly in case of buffaloes. Buffaloes, invariably a buffalo which is having RFM definitely turns into the uterine infection. So these are all the factors, managemental factors that can be used to reduce the uterine infection. And we have to understand this infection, which is the organism that causes. I'm not going to explain much, but here these are all the things. E. coli, Archaeobacterium pyogenes, Physophorus necrophorum, and Provetula. These are all the species, they are important. And why I am saying all these things is, in case if some of the antibodies are not responding in a particular area, it is better go for organism isolation and identify what are those organisms. Because mostly the uterine infection therapy and other things are designed based on keeping these organisms in mind. This is the normal phenomena. Once the bacteria is getting invaded, this is mainly for the students, bacterial invasion takes place. And there are some pathogen associated molecular patterns. And I'm taking an example of E. coli here. So LPS is the PAMP for this particular bacteria. So LPS lipopolysaccharide and its binding protein, they combine together. And then these pattern recognition receptors, they are in the uterine endometrial cells. They recognize, for example, TLR, CD14 and these recognize, and then they induce the pro-inflammatory cytokines and chemokines. And these directly attract the neutrophil and then automatically it is clear. Or otherwise, it also increases the acute phase proteins from the liver and then antimicrobial peptide like beta defensin and other things. Then automatically it is clear. This happens when the animal is very clear. So having said, this is the way in which it develops, how it can be first diagnosed, because based on this method only we can identify the infection.